So good afternoon. So thank you for giving me a chance to present our data. And I'm very honored to collaborate or to contribute to the mutagraphs. So because yesterday we had several talks about the signature 16, so I changed my title of talk, and I will briefly present our previous studies regarding to the signature 16 in liver cancer. So our group participated in the ICGC. So our group from Japan analyzing the 3 different cancer types like liver cancer, gastric cancer, and biliary cancers. So, but today I will show our data mainly about liver cancer. And as you may know, that liver cancer is very unique because hepatocellular carcinoma is associated with virus infections mainly. So it is virus infection, hepatitis virus infection is a major risk factor of this cancer type. But we also know that there is several different epidemiological factors, risk factors, including alcohol intake or obesity or other metabolic diseases. So it is also associated with lifestyle conditions. And also it is well known that the liver cancer is more frequent in the East Asian countries and compared to the Western countries. So before we first collaborated with Ludmilus about his first comprehensive analysis of human mutation signatures in human cancers, we tried to understand regarding liver cancer, is there any difference of mutation patterns between different ethnic groups? So this study we started more than 5 years ago because we have Japanese samples, but we don't have any other ethnic groups. So we discuss and we collaborate with TCGA, United States groups, to join their liver cancer data together with our Japanese group data. So at that time, we collected more than 600 liver cancer genomes, mainly exome data, and to analyze difference of the mutation signatures between different ethnic groups. I'll show briefly. This is just PCA analysis, but using this analysis, we compared to the association between the mutation pattern and different ethnic, different clinical backgrounds, and we found, as you see, that mutation patterns are significantly associated with ethnicities. In the left part here, so Japanese and Caucasian are significantly different mutation patterns, even they have the same tumor. And also we found, surprisingly, gender is also associated with these patterns. And also surprisingly, virus, as I said, hepatitis virus is very strong risk factors, but type of virus infection does not associate with mutation pattern, suggesting that virus itself does not induce specific mutation signatures, but ethnicities or gender has some association with different mutation patterns. And using the mutation signature extraction, at that time we used only exome data, and it is very previous, this very immature studies. But at that time, we could extract 3 unique signatures in a cohort. This is signature 16, we discussed a lot yesterday, and signature 22, we also discussed, we all heard from Steve's talks that this is associated with alicylic acid, and signature 1, this is very common cancer signatures. And in a cohort, we found that signature 16 is more frequent in Japanese and also male patient. And the signature 22 is alicylic acid signature is more frequent in the Asian and Chinese or TCG Asian samples. And signature 1, this is common in all groups. So this data suggesting that the Japanese male patient has a very unique preference of signature 16 compared to the other groups. And at that time, we don't think that alcohol is associated with signature 16 because we know that Americans also drink beers. And because, as I said, that the germline, gender is also associated with difference of signature. So we don't think that any SNPs may associate with this difference of signatures. But now we know that there is, as we know that there are several papers suggesting that 16 is associated with alcohol intake. So also, we compare to the driver gene in different ethnic groups. And this is Japanese cohort, and this is a little it's a French studies. But we found that there is not much significant difference of driver genes of liver cancer between different ethnic groups. So even there exists a significant difference of mutation signature, mutation processes, but the driver gene is not so much different among the different ethnic groups. That is our main conclusions of these studies. And then we launched the whole genome sequence in 4 years ago with Japanese cohort in more in-depth analysis of the signature in liver cancer samples. And we analyzed, at that time, we collected 300 Japanese liver cancer samples, and we did whole genome sequencing, and we tried to extract de novo extraction of mutation signatures in this cohort. And then we found 7 different signatures in the cohort. Actually, this is 3 years ago data, so now we have to use a signature profiler to precisely annotate the signatures, but we found several, like signature 1 or signature 16 here, so several signature 16 here, so several known signatures in our cohort. And then, based on these signatures, we tried to classify our Japanese 300 cases of samples. And here is each signatures we extracted, and we clustered them based on the contribution of these signatures. And we found that there is some signatures are more broadly implicated in Japanese cohort, like signature 16 is here, so it's more than 40% of cases has much contributed by signature 16, and others are also contribute some portion of groups. And also we found a very small group of patients which is highly enriched in specific signatures. So we don't know the cause of these signatures, but suggested that probably these patients may be exposed or some taking a very specific carcinogens which could be different from other Japanese patients. So understanding these cause of signatures may understand the additional risk factor of this liver cancer in Japanese cohort, but also probably in other countries, other ethnic groups. And we tried to correlate with these clusters with non-clinical backgrounds. And this time, we found that signature 16 dominant groups are significantly associated with alcohol intake. So probably, because we did the whole genome this time, so we have more precisely evaluate the contribution of each signature. Then we found a significant correlation with specific epidemiological factors and signatures. And also, there is some portion of patients which has signature 4, which is associated with tobacco smoking, and exactly this group is significantly associated with smoking. And also, these groups are also associated with TAT promoter mutation, which is one of the major driver genes. So some signatures are associated with specific driver genes in liver cancer. So these are our previous studies. So using this large Japanese cohort with other ethnic groups, we think that signature 16 is very unique signatures in Japanese liver cancer cohort. So next, I will show some additional data about epigenetics, association between epigenetics and the mutation signatures. So this analysis says there is a diversity of signature contribution among patients, but I also have a question about is there any different distribution of signature through the genomes or cancer genomes? So of course there is several studies already shown that genome topology is significantly influence the distribution of mutation signatures, such as replication timing or nucleotide occupations associate or influence the distribution of mutation signatures. But we also think that probably DNA modification and DNA repair is importantly contribute to the generation of mutation signatures. So such kind of a DNA adduct formation was as a DNA assault, and repairing these modifications, the mutation is introduced in genomes. But I think that these processes are also influenced by chromatin structure. So I would like to know, is there any specific difference of the distribution of mutation signatures around in the epigenetic context? So using our liver cancer data, we tested this hypothesis. So to analyze the chromatin status, we use NIH roadmap data to define the chromatin status through the genomes. Also, we use these data to annotate each genomic regions, and then we try to examine the correlation between the mutation signature and these different chromatin status. And we found that there is one example, but the signature 16, as I said, this is very unique in the Japanese cohort, and also recently we heard this is also very frequent in Japanese esophageal cancer samples, more significantly enriched in active chromatin genomes, and also transcription start sites. So this signature is very specific, unique target regions to happen. And so this is one unique data. And also then we separate the genome based on the chromatin data, and the one is the active chromatin, so it's more open chromatin, and this is inactive chromatin. And then we compare to the number of mutation signatures between the active and inactive protein. And again, this is signature 16. This signature 16 is significantly and most highly enriched in active chromatin compared to other signatures. We also found there is additional signatures which is enriched in the active one, and also in livers, the other signatures are enriched in inactive chromatin. So this is just liver cancer data, but we speculate that probably chromatin status also influence the distribution property, the generation of the different mutation signatures in human cancer. So this is just a last summary. So as I said, that there is a different chromatin status, and closed chromatin, inactive chromatin prefers several signatures, including signature 1 and 5, and signature 16, and others are more enriched in active chromatin genomes. And I don't show data today, but virus genome integration is also very important in liver cancer carcinogenesis. And we found that also that the B virus genome integration more frequently occurred in active chromatin status. So this mutation process and also virus integration are influenced by the epigenetic status, chromatin status. So this is my summary. So I show that there is a distinct contribution of mutation signatures across different ethnic groups, but there's not much difference of driver gene. So suggesting that different mutation process, but they targeting the same driver gene in the same tumor type of the different geographic regions. And secondly, we using the large-scale Japanese cohort, we found there is a characteristic combination mutation signatures, and also small groups, small patients, which has highly dominated with specific signature, suggesting that there is additional unknown unique cause in liver cancer. And also lastly, I just showed some preliminary data about the association between chromatin status and the distribution of mutation signatures, suggesting that probably epigenetic feature may impact on the distribution or frequency of the mutation signatures, and these probably may help to understand the underlying mechanistic process of this carcinogenesis. Okay. So this is my acknowledgement. So thank you very much for your time.